We'll go ahead and get started. This session is recorded for your viewing purposes later. So we will have this posted to our user support hub in the next week or so. So those of you who did probably thinking, oh, I want to see this recorded, we will have it posted soon. So I have a few announcements and then we will introduce our speaker for today and then we'll get started. But just for reference, my name is Sam Stewart. I'm a project manager here at the All of Us Research Program at the Data and Research Center. I'll be your host today that will be helping moderate any questions or inquiries about our presentation. I do want to make a quick announcement for those of you that are interested. So the University of Arizona Banner Health is seeking seed grant applications for research projects that use the All of Us Researcher Workbench. So they are looking to fund up to 3 proposals with grant opportunities of up to $40,000. So these are open to anyone who's in the United States that has a data use and registration agreement in place. So if you are interested or think that applies to you, please scan the QR code to learn more about the application process. So now I'm going to introduce our speaker today. We have Dr. Matthew Solomonson. He's an Associate Director of Genomic Data Visualization at the Medical and Population Genetics Program at the Broad Institute. The Broad works very closely with the All of Us Research Program, so we're really excited for him to speak with us today. He is a protein structural biologist and software engineer. Matthew is interested in developing customized web application and software tools that enable researchers to explore large, complex biological data sets on the web. I believe he helped work on GeneBAS, which I know many of you are familiar with. I know I get a lot of questions from researchers about GeneBAS, and he has been one of the lead people on our All by All browser, which he's going to give you a preview about today. So our team is very excited to introduce him. And Matthew, I will pass it on over to you.
All right, thank you so much, Sam. I'm really excited to be here and show you our work on the browser. So, oops, okay. So this is the All by All browser, a screenshot. So I'll be going over this resource today, mostly like a walkthrough of the user interface and how we intend people to use it. Before I get into that, I just wanted to recognize the contributions from the team we have at the Broad Institute. We have we are operating within the translational genomics group at the Broad. We have a really talented team of people, basically two full stack software engineers, Phil and Riley, a data engineer named Josh, myself and Steve. We are responsible for leadership and management of the group. And then we often take on co-op students. Currently, Alyssa is with us this semester. We also maintain a wide range of genomic data sets. I just thought I'd mention the most notable probably is the Nomad browser. It's a data set of hundreds of thousands of exome and genomes with a wide range of variants, like short variants, structural variants, STRs, mitochondrial variants, and all the analyses we do on top of that. We also maintain a resource called Genie for estimating allele carrier frequencies and genetic prevalence. We maintain a bunch of internal tools for curating like things like loss of function variants. We also have a whole suite of disease-focused consortia browsers for many of the projects the Broad Institute is focused on, like schizophrenia, bipolar, autism, epilepsy, IBD. A few years ago, we were also involved in the COVID-19 host genetics initiative, which was this really cool effort where we had 3,000 scientists across 120 research groups doing a really big COVID GWAS. And then we also, as Sam mentioned, we developed GeneBAS, which is the resource most similar to the All by All resource. And this is for browsing gene and variant associations in the UK Biobank. Right. So this slide basically showcases the breadth of the data that we have in the All of Us project. The top row shows the phenotypic data, capturing the participants' health, lifestyle, clinical information. And in the bottom, we have all the genomic data, like detailing the genetic variations. So together with these data sets, we're positioned to conduct these large-scale phenotype phenome-wide association studies, or PheWAS, linking these genetic variants to all the traits. So here, this is sort of describing the processing of the phenotype data in the project. On the left, we have kind of how the sample set was arrived at using ancest various age, sex, and relatedness metrics. We filtered that down to a data set of 245,000 samples across the six continental ancestries. On the right-hand side, we have the phenotypic data, both binary and quantitative traits. In the binary category, we have various prescription drugs, family health surveys, fee codes, and on the right, we have the quantitative traits of 7 physical measurements, of which 3 custom phenotypes were derived, and then 60 lab measurements. And then together, these come together to create basically a phenotype manifest of around 6,600 phenotypes. On the top here, we have a breakdown of the phenotypes across the 6 ancestry groups. And on the bottom, we have the number of phenotypes defined across the various ancestry groups. So this resulted in about 3,400 unique phenotypes across the ancestries. And so the phenotype and genomic data went into this pipeline, which was carried out by Conrad and Wenhan. This basically resulted in a series of matrix tables, Hale matrix tables for the gene results, the exome variant results, and the genome results. And then from those, a meta-analysis was carried out. And all this was possible using the Hale batch pipeline that we have at the Broad Institute. About 8,895 GWAS and rare variant association tests were carried out. There's about 2.7 million jobs or 845,000 CPU hours. So a lot of compute going on here. This resulted in 18 ancestry-specific matrix tables and 3 meta-analyses tables. All of these are available on the researcher Workbench. So that result, like across all the phenotypes and ancestries, you basically get 36,936 individual phenotype result tables and 21 Hale matrix tables. So there's a lot of data there to go and browse. I think totaling almost 500 billion associations. And using these pre-computed resources users are gonna find that the costs are significantly lower. So I'd encourage you to go to the Workbench and look for the phenotype of interest. There's an article here on the user support hub that you can go to sort of see what this is, or how to use all the all by all tables. And then there's a whole suite of workspaces for working with the data as well to look at. And yeah, I'd also encourage you to go check out these talks previously by Wenhan, especially on the association analyses, and Amon on the phenotype curation process. So now getting to the actual browser that we developed, you can find it at allbyall.researchallofus.org. This is a screenshot of the homepage. So it's using these 3,417 phenotypes. We have both the gene and single variant data available across 215,000 participants, both exome and genome sequencing data. And it currently displays results from the meta-analysis. This is a general overview of the navigational scheme of the browser. So on the top bar, we have the various links that you can click on, and then we have a status bar below that, which shows you basically like the active phenotype, active gene and region. So keep in mind when you're clicking through, like this will show you what is currently active. And then the bottom part, we split the page into a phi-wise Manhattan plot, QQ plot view. So this is kind of like where we keep all the data set overviews. So what are all the associations across a given gene or phenotype? And on the right-hand side, this is sort of like the zoomed in view. So what are the gene burden statistics and variant associations for kind of like a single region in the genome? And then there's controls on either side for the phenotypes and for the variants. You can click on the middle and drag it back and forth, depending to make the real estate bigger for whichever type or whichever side of the screen you want to look at to focus on. So there's the actual screenshot of the navigation. Like I said, this is sort of the big picture view on the left-hand side, and then the zoomed in view on the right-hand side. In general, the associations will load more quickly with sort of the stronger associations. So if you see anything in the browser with the green and orange circles, like those will generally load more quickly than the non-significant associations. This helps us basically save on like compute costs. Um, this is the phenotype page. So this is how you can explore associations for a particular phenotype. Looking at cholesterol here, you get there's a little menu to jump to various sections within the phenotype page. So you can look at the gene Manhattan plot or the variant plot. Then there's like information about the phenotype, like the identifiers, number of cases and controls, that kind of thing. And then we have the Manhattan plot, QQ plot, and then the table below. So this is an ordered list of all the genes associated with it. And then below that, even further, you can see the variant association Manhattan plot. So clicking on different genes in this list will update the right-hand side of the page. So it gives you a really quick way to browse through all the hits. There's 3 different gene burden test types available in the resource. We have burden, scat, and scat-O, each with their different types of trade-offs. We have 3 burden sets as well, P-LOF, missense, and low confidence LOFs, and synonymous. So this is all displayed on a gene page. We have the 3 different p-values as columns, and then as rows we have the different burden sets. So for any given gene phenotype pair, you can view that table to get all the detailed statistics. Then we have the PheWAS plot, which is for looking at all phenotypes for a given gene. So this actually shows the top. We have 2 views. We have on a per gene basis, and we also have the top genes in the whole data set you can look at. So this has various controls like filtering for phenotypes by keyword, selecting the burden tests and burden set. You can also filter by category. And then, yeah, there's different controls also for controlling the type of plot. So you can look at the p-value plot, the beta plot, or both of them. This is the view for single variant associations plotted along the gene. They're basically plotted along the exons. There's different controls on the right-hand side, like for selecting the type of burden analysis to show. You can see the allele frequency scaling plot, legend, sorry. And then there's different columns you can toggle on and off depending on what you're interested in. You can use these controls. We also have features for browsing the GWAS results, like the single variant associations. So these will display everything in a Manhattan plot. Clicking on the peaks or on the arrows in the table will update the right-hand side of the page to zoom into those particular regions so you can get a more detailed view. And then this is like kind of what it looks like to zoom in on the locus, and you can kind of expand the whole page so you can see the whole thing more clearly. It's got the genes plotted along the bottom, and we also show all the gene burden results in the region in a little table here. So with that, I will hop into a demonstration with the actual browser. Guess that's not a real link. Okay. So here we are at the home page. So I can start by searching for like a gene. So I can type in LDLR. I'll get a list of all the genes that match the query. Then I get all the gene identifiers. I'll just make this a bit bigger so people can see. We also have nomad gene constraint here, so that's kind of helpful when you want to for cross-referencing with gene burden results. We have links out to various resources, including the the All of Us data browser. You can link to Nomad. We also have a link to GeneBAS, so you can compare quickly like the associations in this resource. And we have a UCSC link too. And then going down into the PHWAS plot, uh, um, so yeah, we have the all the associations plotted here at a negative log 10p scale. I can jump between the different burden results. Um, I can change what's being displayed in the plot, like beta or both beta and p values. I can change to the missense data set or the PLOF. So I jump back and forth. You can see once I load something once, there's caching happening on the client side. So it should be quicker to look at, certain phenotypes or genes a second time. I can also click on the categories uh, and shift click or command click to kind of look at groups of categories that I'm interested in. And that'll just filter the table and the plot to those categories. And then if I want to dig into particular phenotypes, I can click on this little arrow, which will then pull up the detailed associations for this gene. I see the three or the matrix of p-values and burden sets at the top. I can look at the individual variants here. I can filter to different classes of variants. So it's just a way to zoom in on what variants were actually included in these burden tests. Yeah, so I can click on different column presets. Some of these are more useful for binary versus quantitative traits, or just looking at the general population level frequencies. Right. So then now I can go into a particular phenotype of interest. So I'll use the search bar to, for example, search for cholesterol and go to that. So I have phenotype ID, description, what is it, continuous or quantitative, or sorry, binary, um, number of cases, stuff like that. I can see the gene Manhattan plot and the QQ plot for genes as well. I can click here to look at the details of each gene, just like I could with the PHWAS section. So easily go up and down this list and look at the details. I can also hop down to the the single variant Manhattan plot by pressing that button or jump around pretty easily like that. So this gives me a classic Manhattan plot with all of the variants in the table here. I can also filter to different classes, uh, a variant to missense, LOF, synonymous, other. I can search for a variant by I think anything in the table, basically any text field should be searchable here. I can also zoom into particular chromosomes using this dropdown. So if I want to look at these hits on chromosome 19, I can select that. And now I see a sort of a zoomed in view of those two peaks. I can also like click on those peaks to look at them in more detail. So I can kind of drag things around to make more real estate. So yeah, so now we're zoomed into this region of chromosome 19. In the gray, we sort of have these are like the non-coding variants. In red, we have the PLF, yellow missense. So what happens if yeah, so I can kind of focus in on different variant classes that way. Um, Down here, we have all the genes in this region as well. So I can click on any of them to like investigate any of them further. We also have, I'll just go to this locus because it has a better demonstration of this, but this is going to get the LDLR region. So now I can go down here, look at the table and see all the burden statistics for the genes in this region and see, yeah, LDLR, not only does it have this common variant signal in this particular locus, but it also has, strongly significant gene burden results for both the PLOF and the missense variant burden sets. So that's convincing evidence that, LDLR is underlying this association. All right, so what else? There's a walkthrough if you need more detail about this. Um, oh yeah, you can also like click on genes and sort of, yeah, zoom into the gene, look at what's going on here, and then kind of jump back like that. So there's a way to sort of just investigate all of these. So I'm getting a lot of light happening in my house. Okay. Right. Right. So there's a walkthrough for like a lot of what I just talked about. You can go here and um, see, most of this talk is kind of just typed out like this. So feel free to learn more at the walkthrough. Um, yeah, I think that's about it for the walkthrough or the demo. I'm curious if anyone has like any feedback about the resource, like what do you think about it? What do you wish it could do? I'd be just interested to know like um oops, that shouldn't be happening, but what do you think? So thank you.